dear viewers, welcome to my channel BioQuest Biochemistry classes. Today I am back with a video tutorial on hot air oven and autoclave. Let us start today's session with hot air oven. It uses dry heat to sterilize objects in the laboratory like glassware and metal equipments. This hot air oven is also termed as forced air circulating oven. This instrument has been developed by Louis Pasteur. It works on a wide range of temperature, and the span of temperature may be 50 to 200 degree centigrade. The working principle of hot air oven. The electrically heated oven that is fitted to a fan causes equal distribution of hot air, resulting in heating of external surface of articles followed by transfer of that heat towards the center by conduction. So, it utilizes dry heat that is conducted from the external surface to the center of the objects that need to be sterilized. This hot air oven is also fitted with a thermostat that maintains. Let us check the parts of a hot air oven. This one is the thermometer that will record the temperature. This hot air oven is made up of a double-walled insulated chamber, and it is either made up of aluminium or stainless steel, which resists mechanical shock or oxidation. In between this double wall there is presence of an insulated layer of glass wool. Inside there are perforated aluminium tray where articles that need to be sterilized are placed. Here you can see there are a panel of knobs which contain the main on or off switch, heat regulator knob. This is the direct through timer and temperature regulator knob, or this is the timer, and this is an indicator which indicates that the red light, it will indicate whether it is working or not. Now this is the door of the hot air oven. This door, it is attached to heavy hinges. The presence of heavy door gasket on the side is used to reduce heat loss during operation. This gasket, it is made up of asbestos and it secures that no heat is lost during the operation of hot air oven. Let us check the common types of hot air oven that are utilized in our laboratory. Natural convection oven, forced convection oven and side draught oven. Most common type is the forced convection oven. Let us move on to the standard operating procedure of hot air oven. Place the articles on the aluminium trays or shelves and do not overload the trays or shelves. Next, close the door firmly so that no heat is lost during the operation. Switch on the main switch. Set the temperature by regulating or adjusting the knob. Keep it on for the required time. Just set the timer how long you need to sterilize the article. Once the required time is reached, turn off or switch off the hot air oven. Remove the articles with thermal gloves or tongs to prevent any sort of thermal or burn injury to the person operating the hot air oven. Now, what are the applications of hot air oven in our laboratory? It is used to sterilize glassware. It is used in heat treatment and drying of samples such as metals, alloys, and in the biochemistry lab it is used in thin layer chromatography or chromatographic separation of carbohydrates in development of the color. Let us move on to autoclave. We have seen hot air oven works on the principle of dry heat for sterilization. Reversely, autoclave works on the principle of moist heat sterilization. What does that mean? It operates at high temperature, that means it uses steam and high pressure in order to kill microorganisms and spores. The high pressure facilitates deep penetration of heat that results in coagulation of proteins, which causes irreversible loss of the proteins and damages the microbes. The water boils at 121 degrees centigrade at the pressure of 15 pounds per square inch for 30 minutes, and that steam is used for sterilization of articles. So, autoclave requires water that need to be converted to steam at a high pressure, and that steam is used for sterilization. This steam destroys the microorganisms and spores by giving off latent heat. If you recall our basic physics of school days, we have seen that steam causes more thermal burn or injury in comparison to water because it has higher content of latent heat. Let us check the parts of a autoclave. This is the simple pressure cooker type of autoclave. Here you can see there is a gauge which is used for measurement of the pressure. This is the pressure valve and this is the safety valve. There are two outlet valves. One is for releasing the steam, another is for releasing the vacuum. And these are the body of a normal type of autoclave that is made up of stainless steel. Let us check the standard operating procedure of autoclave. Put sufficient amount of water inside the chamber. Place the articles that need to be sterilized inside the chamber. The lid is then closed and screws are tightened to ensure an airtight condition. Now switch on the autoclave. The safety valves are adjusted to the desired pressure in the temperature. Routinely 15 pounds per square inch of pressure is used. As the water inside the chamber boils, the air-water mixture is allowed to escape through the valve to be displaced with steam. Initially it is a mixture of air and water. The complete displacement can be ensured once the water bubbles cease to come out from the pipe. The valve is then closed and the steam inside is allowed to reach a pressure that is 15 pounds per square inch in most of the cases. Now the whistle blows to remove the excess pressure from the chamber after the desired pressure is reached, and the autoclave is run or operated for a holding time of 15 minutes in majority of the cases of sterilization. Autoclave is switched off and allowed to cool until the pressure gauge indicates that pressure inside has lowered down to that of the atmospheric pressure. Next, the lid is opened and sterile materials are removed. So, this is a simple SOP of autoclave. Now, let us check what are the different variants of autoclave that are used. Pressure cooker type, you have seen the figure in the previous slide. This is the most common variant. Horizontal autoclave, vertical autoclave, and common laboratory autoclave. In some of the cases, you can see that the common laboratory autoclave, it is a benchtop device and it looks like a microwave that is used in day-to-day purpose in our kitchen. So these are the variants of autoclave. Now, what are the applications of autoclave in our laboratory? It is used to sterilize culture media, pipette tips, and it is also used to sterilize biomedical waste prior to disposal. It is mandatory that majority of the labs should have the provision of a dirty autoclave or waste autoclave sterilization, that is known as a dirty autoclave, and to check whether the autoclave is functioning properly or not, every lab should have a protocol to check that using a chemical indicator and biological indicator. The use of autoclave pertinent to biomedical waste has been given in a video tutorial in my channel on biomedical waste, the link of which is given in the I button above, or you can check the description box. So, these are the uses of hot air oven and autoclave. Hot air oven uses dry heat for sterilization. Autoclave uses moist heat for sterilization. In hot air oven, it works on a wide range of temperature as high as 200 degrees centigrade. For on the other hand, autoclave, they work on the principle of moist heat that utilizes steam as well as high pressure for sterilization. So these are the basic differences between autoclave and biomedical waste. If you like my channel and find it useful, do like, share and subscribe. Thanks for watching.